For decades, scientists have been experimenting with the idea of changing the human genome to cure genetic disease. Some researchers now say they are closer than ever before to developing a treatment to correct defective human genes thanks to new gene editing technology that could provide scientists with unprecedented power to rewrite the code of life. With the new technology called CRISPR-Cas9, scientists hope to create future medicines that can fix errant genes that cause diseases such as cystic fibrosis and certain cancers. The process involves two basic components, the Cas9 protein and a programmable so-called guide RNA. The guide RNA is a single molecule that directs Cas9 to the specific sequence in the genome. Once there, Cas9 works like a molecular scalpel, cutting out the flawed part of the sequence that causes the mutation. A gene-editing drug would include the Cas9 protein and a guide RNA specifically targeted for the mutation. It could also carry healthy DNA to fill in the gap. Scientists say CRISPR-Cas9 is a revolutionary tool because it allows them to target specific genes more precisely than previous gene correction technologies. There are other advantages as well.
The nice thing about Cas9 is it's very easy to use and very inexpensive to use. It doesn't take you years and years and millions of dollars to try your idea. It takes you weeks and a couple of $100s. And so that means that you can be very creative and very broad in the things that you test.
Scientists believe the new technology could find many applications in biomedical research and eventually allow them to create treatments for diseases caused by mutated genes. It may be years until the first human clinical trials are conducted and maybe a decade or longer until gene-editing therapeutics hit the market. But many laboratories and pharmaceutical companies across the country and the globe have adopted CRISPR-Cas9 since it was first introduced in 2012. Some pharma companies are already exploring how to create gene-editing drugs for certain genetic diseases. Some labs are using the genome editing technique to genetically engineer laboratory animals with genetic mutations and then study the recreated mutations inside the animals. At the Innovative Genomics Initiative in Berkeley, California, scientists recreate diseases in human cells to learn more about how mutations cause illnesses and how they can be corrected. Although many scientists have high hopes for the technology, there are still challenges, they say. For example, CRISPR-Cas9 is generally precise, but it occasionally cuts the genome at unintended sites.
I mean, we're talking like 1 in a million, 1 in 100 million. So Cas9 might come in and it might make a cut, and it might also make a cut somewhere else. And we're still trying to understand how frequently off-targets occur, how much specificity is a problem. As you can imagine, from a therapeutic point of view, we want to know the answer to this.
Scientists say it's also difficult to deliver genetic changes to all the cells in the body that need it. To treat some diseases, scientists will be able to extract blood cells or bone marrow, alter them, and then put them back in. For diseases in more complex regions, like neurological diseases, more research is needed to find out how to get the Cas9 protein to the right spot. There is still a lot more work to be done, but many scientists believe that the Cas9 technology could one day help cure many genetic diseases.